Check, will you, ma? How many pages we have studied to this moment?
We're on page 24, sir.
Page number 24, correct? We have already entered protein, correct? Primary, secondary, tertiary. Tertiary structure, did I tell you?
No, sir.
No. So I have stopped at secondary structure. Is it so? Have I stopped at secondary structure?
Yes, sir.
So if you take the tertiary structure will be appearing like a spherical part. Okay, it will be appearing like a spherical part. Okay. In the tertiary structure, there will be additionally 4 types of bonds. If you take the tertiary structure of protein, so this is how the tertiary structure will appear. It is described by the word tertiary structure, a single polypeptide chain folds, coils, and finally make a single unit. So how many polypeptide chains involved in making the tertiary structure? How many polypeptide chains involved in making the tertiary structure? Look at the diagram. How many chains are here? How many chains are here?
4.
4 chains. Can you see the diagram? Do you see 4 chains there? Do you see 4 chains here? How many chains are here?
1 chain.
Only 1 chain. Within that chain, there are many interactions. One called hydrogen bond. In the tube you have in the hands, do you notice hydrogen bonds?
Yes, sir.
Yes. Further, disulfide bonds. Do you notice disulfide bonds?
Yes, sir.
Third, ionic bond. Is there ionic bond?
Yes, sir.
Fourth, hydrophobic interaction.
Yes, sir.
Now, in last class lesson, Did I introduce you amino acids? In the last class lesson, did I introduce you amino acids? No.
Yes, sir.
This side chain, this part, did I introduce you? No. No, no. Yes or no?
So you introduced the backbone and chitin, that things.
I introduced you backbone and side chain. Yes or no?
Yes, sir.
Yes. So that is very important here. Here, ionic bond, hydrogen bond, disulfide bond, hydrophobic interaction. All these four are created among the side chains. Understood? Understood? Did you understand what I told you? Hello?
So can you repeat it?
All these interactions are created between the side chains. Now see, this is a side chain. This is another side chain. Between side chain and side chain, what bond is created? What is
Ionic bond.
ionic bond? Similarly, this is a side chain. This is a side chain. Between side chain and side chain,
Hydrogen bond is created.
hydrogen bond is created. Same to the disulfide bond. It is also created between side chain and side chain. Hydrophobic interaction is also created between side chain and side chain.
chain.
Remember, in the secondary structure, go to the secondary structure. In the secondary structure, what is the specializing bond? What is the specializing bond in the secondary structure? In the secondary structure, the specializing bond is hydrogen bond. What is the specializing bond in the secondary structure?
Hydrogen.
Hydrogen bond. That hydrogen bond is created between the portions of the bond is created between the portions of the
Backbone.
backbone. Okay, so secondary structures hydrogen bond, tertiary structures hydrogen bond. Both are hydrogen bonds, but in the secondary structure, the hydrogen bond is created between the components of the
Hydrogen.
in the secondary structure, hydrogen bond is created between the components of the,
Oxygen and hydrogen.
that's not necessary, components of the
Backbone.
backbone. In the secondary structure, hydrogen bond is created between the components of the backbone. That is true. Oxygen and hydrogen. Okay. in the tertiary structure, any of the bond is created between the side chains. Side chain and side chain bonding by hydrogen bond. Side chain and side chain joining by ionic bond. Side chain and side chain joining by hydrogen bond, disulfide bond, hydrophobic interaction, all these. Are you clear? Good. In the tertiary structure, these examples are there. Read these examples.
Enzymes, myoglobin, albumin, immunoglobulin.
Enzymes, myoglobin, albumin, immunoglobulin. Later, in future lessons, you will study a clear idea regarding each of these components. Enzymes, a full detail there. Myoglobin, you have details about them separately. Albumin, immunoglobulin, separate. Details are, plenty of details are there. But at the moment, you want to know these ones as examples for tertiary proteins. Are you clear regarding tertiary proteins?
Yes, sir.
Are you clear? Good. Coming to the next idea. What kind of structure? Quaternary structure. What kind of structure? What kind of structure?
Quaternary structure.
Quaternary structure. If you take quaternary structure of protein, the very important portion it is, in the quaternary structure of protein, there will be more than one. Keep in the mind. Okay, take this. What is written in the right side? What is written in the right side? Association of two or more polypeptide chains with each other. In the diagram, how many polypeptide chains are shown to you?
Two.
Two. There are instances. Here, how many polypeptide chains are there?
4.
4. Here, how many polypeptide chains are there on the left side?
4.
Left side, on your left side. Collagen,
3.
3. So, 2 there, 3 there, 4 there, more than 1 there. It is the only protein structure having more than 1 protein polypeptide chain. Understood? How many chains are there in the primary? How many chains are there in the primary?
1.
1. Secondary?
1.
1. Tertiary?
1.
1. Here only you have more than 1. Clear? Are you
Yes, sir.
clear? Good. If you take one component here, this one component is called one subunit. So every polypeptide component making the quaternary structure is called by the name subunit. So in hemoglobin, right side picture shows you the hemoglobin. In hemoglobin, how many subunits are there?
4.
4.
Four.
In collagen, how many subunits are there?
3.
3. Between one subunit and another subunit, the attraction will be there. This attraction is called intermolecular attraction. What kind of attraction?
Intermolecular attraction.
Intermolecular attraction occurs between one subunit and
Another subunit.
another subunit. Same time, within the subunit, there will be hydrogen bond like intramolecular attraction. If it is occurring inside the molecule, how will you describe it? If you inside the molecule, inside one subunit, inside mean Baba, we have to call it intra. Understood? Clear?
Yes, sir.
Inside mean, what is it?
Intra.
Intra. Okay. Between mean, between mean,
Inter.
inter. Are you clear?
Clear, sir.
Right. So, please go through the quaternary structure notes and tell me whether you can understand it.
Understood, sir.
Okay.
Okay, sir.
Right. We are coming to the heading called, what is it? Denaturation of protein. What is the heading? What is the heading? denaturation of, what is it? Protein. Right? If you take all these diagrams, one you can notice a protein molecule is losing its structure. Correct? A protein molecule is losing its structure. And that is actually called denaturation. Normally, in a protein molecule, there will be a three-dimensional structure. What kind of structure? What kind of structure?
Three-dimensional structure.
Three-dimensional structure. Due to certain agents, that three-dimensional structure is lost. Losing the three-dimensional structure in a protein is called denaturation. Understood? Understood?
Yes, sir.
When denaturation occurs, the protein molecule will lose its biological function. Its biological function also will be lost. If an enzyme undergoes denaturation, thereafter it can't do catalytic activity. If hemoglobin undergoes denaturation, then it can't transport oxygen. If myoglobin undergoes denaturation, it can't store oxygen. So, during denaturation, the protein molecule is losing its biological activity. Understood?
Yes,
Understood? Right. Right. Who can cause denaturation? So what is denaturation? Please read this one. This is the definition for denaturation. What is denaturation? Read, will you?
Denaturation of protein is the loss of specific chemical three-dimensional shape due to the alteration of weak chemical bonds and interactions.
Loss of specific chemical three-dimensional shape. If a single word is missed, no marks. Keep in the mind. Understood? These kind of things you will be getting 1 or 0. I am not here to tell you to make you fear, but that is the truth, Puta. That is the truth. Okay. If a single word is missing, you will be completely losing the marks on definition. Right? So that's why from the beginning, I have told you to compile the definitions. Practice the definitions. Okay, those things are told to you because a single word missing. Okay, we can't go and argue. Teacher, I wrote it, no, only one word missing, no. Give me half marks, mean no. Understood? Understood?
Yes, sir.
Okay.
Yes, sir.
But remember, the profession is so that accuracy is very, very important. Okay. The stream is so. Right. So, alteration of weak chemical bonds and interactions. This is how they have told. Alteration of weak chemical bonds and interactions. Majorly hydrogen bonds, hydrophobic interactions, ionic bonds. They are the three coming under weak chemical bonds and interactions. So who are the weak chemical bonds and interactions? Who are the weak chemical bonds and interactions?
Ionic bonds,
Ionic bonds, hydrogen bonds, hydrophobic interactions. Who are the ones coming under weaker bonds and interactions?
ionic bonds, hydrogen bonds, hydrophobic interactions.
Comparatively, disulfide bonds are very stronger bonds. Comparatively, disulfide bonds are very stronger bonds. So they will not easily get altered. But there are opportunities. They also can get altered. Remember one more thing. Three-dimensional structure is not noticed in secondary structure, or mainly primary structure. Primary structure, are they speaking about the three-dimensional? Are they speaking about the three-dimensional?
No,
No. Look at the secondary structure. Are they speaking about the three-dimensional?
no,
No. Okay. Look at the tertiary structure. Are they speaking about the three-dimensional?
Yes.
Yes. Look at the quaternary structure. Are they speaking about the three-dimensional?
no,
No. But they say in the quaternary, it can be a tertiary protein or a secondary protein. Now, if you take hemoglobin, hemoglobin is the aggregation of tertiary proteins. Then those who have three-dimensional shape only can undergo denaturation. Are you clear? Are you
Yes.
clear? It is the loss
yes,
of the specific chemical three-dimensional shape, no? Correct? Without having three-dimensional shape, can a protein undergo denaturation?
No.
No. Those who have the three-dimensional shape only can undergo
Denaturation.
denaturation. Are you clear?
Yes, sir.
Right. Now,
yes,
when if you take some egg white, egg white?
Yes, sir.
Yes. Egg white is a protein. If you put that egg white in a hot plate, in a hot oven, to what color it will turn? Omelette making. Omelette making. You take the egg white and put on the saucepan. To what color it will turn?
White color.
White color. That is denaturation. That is denaturation by heat. Understood?
Yes, sir.
Yes.
Right. What are the agents of denaturation? Who can cause denaturation? Denaturation can be caused by various agents. One, it is these. Read this one.
High
Read this
temperature and
one.
high energy radiation.
High temperature and high energy radiation, something like UV, ultraviolet. Further, strong acids, strong bases, high concentration of salts, heavy metals, organic solvents, detergents, that means soap items. All these can cause denaturation. Understood?
Yes, sir.
Right? In exam paper, denaturation can come to you as a short notes. If we can write really 20 points, that will become a short notes. If we can write nearly 20 points, it will become a, what is it? Short notes. So this can be a short note. Understood? Understood? Right. So in the follow portion, I am introducing you amino acid. Right? In the last class also, I introduced you. Anyway, now it's an official introduction. Okay? In the amino acid, there are two separate portions you can identify. This portion is called by the name backbone. Last class, did you mark backbone?
Yes, sir.
Yes. Those who have not marked, please mark the backbone. Asma, mark down the backbone. You have the tute now.
Yeah, sir. Okay.
Now mark it down the backbone. Right. Other than backbone, what is remaining is called that R part. That R part is called by the name side chain. Now, Let me introduce you little by little the components. Here I am choosing alanine structure. What is the structure? What is the structure?
Alanine structure.
Alanine. Alanine is an amino acid. Alanine is an amino acid. Can you see this amino acid? Right?
Yes, sir.
In this one, this group is the amino group. This group is carboxyl group. This is carbon atom. This is hydrogen atom. All these four together make the
Backbone.
backbone. Here, this component is the side chain. What is the side chain, Asma? What is the side chain here? Side chain here? CH3. What is the side chain?
CH3.
CH3. So for every amino acid, there will be a different side chain. In all the amino acids, backbone will be same. Understood? Whatever the amino acid you choose, there will be an amino group, there will be a carbon atom, there will be a carboxyl group, there will be a hydrogen atom. Will those portions differ? Will those portions differ? Tell me. Will these four pieces differ from one amino acid to another amino acid?
No.
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 Amino group will be there, carboxyl group will be there, carbon atom will be there, hydrogen atom will be there. The only changing part is whom? The only changing part is whom?
The side chain.
The side chain. Okay, when I show you different amino acids, you should be able to identify different side chains. Understood? This is alanine. I will take another amino acid. This time I am choosing the amino acid called, what's it? Read this name, Puta. Cysteine. Read it.
Cysteine.
Cysteine. Amino acid's name is cysteine. Cysteine. Okay. Let us take this diagram to represent cysteine. Can you see the amino group? Can you see the amino group?
Yes, sir.
Yes.
Yes. This is the amino group, correct? Can you see the carboxyl group?
Yes, sir.
Yes, this is the carboxyl group. That means this is going to be the whom?
Backbone.
Backbone. If that is the backbone, this piece is going to be the
Side chain.
side chain. In the side chain, here there is sulfur atom also, correct? Yes
Yes,
or
sir.
no?
Yes, sir.
Yes. So it's a sulfur containing amino acid. I choose another one. Their names are not fully in syllabus. Okay. Their names are not in syllabus. Just for an idea, I am showing you one by one. You don't need to memorize their structure. Clear? Right. What is this molecule? Read the name of the molecule.
Glutamic acid.
Glutamic acid. In the glutamic acid, this part is going to be the
Backbone.
backbone, carbon, amino group, carboxyl group, hydrogen. Then the rest here is going to be the
Side chain.
side chain. So one amino acid to another amino acid, what is changing? What is the
Side.
component changing? The side chain is changing. Are you clear?
Yes.
Are you clear? Right. Now, take this carbon. This carbon is surrounded by 4 groups. What is this group? Tell me.
Carboxyl group.
Carboxyl group. Who is this?
Hydrogen.
Hydrogen atom. Who is this?
Amino group.
Amino group. This you call side chain. Okay. Are the 4 blue components different? The 4 blue components surrounding the carbon, are they different from each other?
Yes.
Yes. If it is so, that carbon is called by the name, what kind of carbon?
Asymmetric carbon.
Asymmetric carbon. If at least 2 are same, then it will be coming to what kind of carbon?
Symmetrical
Symmetric.
carbon.
Symmetric carbon. In your chemistry, and there's a section called organic chemistry, one of the biggest section in the chemistry lesson. Your chemistry sir will teach you a full detail why it is asymmetric, why it is symmetric, symmetric what it can do, asymmetric what it can do. All those details your chemistry sir will teach you. I am not here to tell those details that will be beyond our limits. Understood? So how can you decide if the 4 groups are different? It is what kind of carbon?
Asymmetric.
Asymmetric carbon. If at least 2 are same,
Symmetrical carbon.
symmetrical carbon. Now I am choosing another molecule, another amino acid. It is, what's the name? Read the name. Read the name.
Glycine structure.
Glycine, glycine. So I am choosing glycine here, right? How to pick that correct picture now? Right. Here, can you see the, so what is this group? What is this group?
Carboxyl group.
Carboxyl group. What is this group?
Amino group.
Amino group. What is this?
Hydrogen.
Hydrogen. This is also
Hydrogen.
hydrogen. Actually, here R is a
Hydrogen atom.
hydrogen atom. That's the truth. Okay. The point here is, can you call this carbon in the middle as an asymmetric carbon?
No.
Can't.
No. Why?
No.
Why can't you call that way?
2 structures are same.
2 structures are same. Hydrogen, again hydrogen, correct?
Yes.
If in that, so glycine is the only amino acid having what kind of carbon?
Symmetric carbon.
Symmetric carbon. All the other amino acids have
Asymmetric carbon.
asymmetric carbon. So only amino acid having a symmetric carbon is whom?
Glycine.
Glycine.
Nice.
Asma, is it clear?
Yes, okay.
Good. Right. Now, let's go to the note. Here we are introducing what are the groups? Amino group. Now you are familiar, correct? Amino group, hydrogen atom, variable group, R group. Do you agree it is variable? Do you
Yes.
agree it is variable? Depending on R, changing the R, how many amino acids are there?
20.
20 amino acids are there. Understood? Understood?
Yes, sir.
Good.
Sir.
Let me come back to this. Okay. If you take glycine or any amino acid, never mind. I'm taking this diagram once again. What is this group? Tell will you?
Carboxyl group.
Carboxyl group. Carboxyl group is an acidic group. Understood?
Yes, sir.
Because it is an acidic group, it can react with, okay, can react with whom?
Base.
Base. It can react with base. Excuse me. Hello? Okay. Now carboxyl group is there. What kind of group it is? What kind of
Acidic
group
group.
it is? Acidic group. Because it is an acidic group, with whom it can react?
With base.
With
Bases.
base. Okay. What is this group? Tell me.
Amino group.
Amino group. Okay. It is a basic group. Base. Because it is a base, it can react with whom?
With
Acid.
acid.
With acid. Clear?
Yes, sir.
Right. So, if you consider overall an amino acid can react with both acid and base. Agree?
Yes, sir.
Do you agree with it? An amino acid can react with both acid and base. If you put the acid, amino group will react with it. If you put the base to it, carboxyl group will react with it. Overall, amino acid can react with amino group as well with, what is it? Sorry. Amino acids can react with acids as well with
Bases.
bases. This nature is called amphoteric. Amphoteric. So amino acids are described by the word, what is it? Amphoteric. Why amino acids are called amphoteric? They can react with both acids and
Bases.
base. Which group will react with acid?
Amino group.
Amino group. Which group will react with base?
Carboxyl.
Carboxyl group. Any amino acid has amino group and carboxyl group, right? Without those two groups, will there be an amino acid? Without
No.
amino group, without carboxyl group, will there be an amino acid?
No.
No. So all the amino acids are amphoteric. Clear?
Clear, sir.
Could you please take a white sheet and mark these details? Tell me after marking it.
Done, sir.
Have everyone completed it?
Yes.
Yes.
Okay. Asma, completed it?
Yes, sir.
Good. Now, I am once again taking glutamic acid. This is an amino acid. How to pick the correct diagram. Aiyayyo. Here, what is this group?
Amino group.
Good. What is this group?
Carboxyl group.
Carboxyl group. What is this portion?
Side chain.
Side chain. In the side chain, can you notice another carboxyl group there?
Yes, sir.
Yes.
So if you take glutamic acid, how many carboxyl groups?
2.
2.
2 carboxyl groups and 1 amino group. 2 acid groups and 1 base group mean which is more stronger? Acidic nature or basic nature? Which is more
Acidic nature.
stronger?
Acidic nature.
Acidic nature. Right? So I want you to mark this diagram in the white sheet, and you have to mark these two as what? Or this one as what? Carboxyl group. That means in an amino acid, they say in the exam paper, they say in an amino acid, always there will be one carboxyl group. Will you agree with it?
No.
No. And amino acids, acidic property and basic property are always same. Will you agree with it?
No.
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 Acidic property can be greater than
Basic.
basic property. Basic property can be greater than acidic property. That will come in another amino acid. Understood?
Yes, sir.
So please mark it down. Keep white sheets always. It's very important. My children, actually, you don't need to memorize these structures. Understood? I am giving these things to you not to memorize. Don't try to memorize these glutamic acid. This is the structure. These things are not for memorization. Understood?
Yes,
That's for an understanding. You should know one amino acid can hold more than one carboxyl group. The side chain can contain carboxyl group. To understand that one. Don't try to memorize them. Tell me after completing it.
Done, sir.
Have everyone completed it?
Yes, sir.
Good.
yes, sir.
Similarly, we are going in search of an amino acid. Okay, this amino acid, it is called Rg9. Did you mark that structure as glutamic acid? That structure you have drawn, did you label it as glutamic acid?
Yes, sir.
Similarly, we are going for another one. What is it? Rg9. If you look for the Rg9, okay, I am looking for a simple structure. Most of these ones are complex ones. Okay, we will choose this. Look at the left side first picture. Okay. Wait. Right. Look at this first picture, left side picture. Right. What is this group?
Carboxyl.
Carboxyl group. What is this group?
Amino.
Amino group. This whole thing is the backbone. Understood? This whole thing is the, what is it? Backbone. Backbone is that one mean. What is this?
Side chain.
Side chain. Agree? Agree. Right?
Yes, sir.
In the side chain, I want to highlight this person. What is this group?
Amino group.
Amino group. So the side chain can contain amino group. Now overall, in this amino acid, how many amino groups are there?
2 amino group.
2 amino groups are there. Okay, if we argue, we can put so many another 2 amino group. That's not my idea. Right. So Rg9 is an example for an amino acid having amino groups in the side chain. Understood?
Yes, sir.
Yeah. So what is this molecule? How do you call this molecule, my children? What is the name of this molecule?
Rg9.
Rg9. So mark it down, the arginine. Mainly, you want to know it has amino group in the side chain. Okay, again telling you, don't memorize the structure. That is not your work. There are so many things to memorize. There are so many things to practice. Plenty of works are there. Unnecessarily don't waste your memory. Understood?
Done, sir.
Have you completed it? Have you completed it?
Yes, sir.
Right. So, with these ideas, if you go here, read this sentence. Read this sentence. Amino acids are
Amphoterate as they
amphoterate.
exhibit alkaline nature by amino acid and acidic nature by carboxyl group.
Good. So here I have listed all the amino acids, but as I told you, you don't need to memorize them. Can you see the arginine here? Can you see the arginine here?
Yes, sir.
Yes, it has. This is the side chain having amino group. This is lysine having side chain amino group. This is histidine having amino group. This is aspartic acid. What is the name? What is the name?
Aspartic acid.
Aspartic acid. If it is finishing with the term acid, you have to understand it has carboxyl group in the side chain. This is glutamic acid. They have written as aspartate, glutamate, but we are reading it as aspartic acid, glutamic acid. Read this one. Read that name.
Methanol.
Correctly read it. Your tute, you have in the tute. Correctly read it.
Methionine.
Methionine. Correct? Methionine is a special one. What is the speciality? Maya. What is the special atom?
Sulfur.
Sulfur. Okay. And here is cysteine. What is the special atom? What is the special atom?
Sulfur.
Sulfur, right. Now, I am asking these questions at you. Certain amino acids have sulfur in their structure. Who are they?
Methionine.
One, it is methionine. Yes. Other one. Other one.
Cysteine.
Cysteine. Correct? Am I right? Right. Some amino acids have more than one carboxyl group. That additional carboxyl group is found in the which portion? Additional carboxyl group is found in the side chain. Who are the examples? Glutamic acid, or we can call it by the name glutamate. Other one, aspartic acid, or we can call by the name, what is it? Aspartate. Some amino acids have more than one amino group. That additional amino group is found in the side chain. Who are the examples? One, it is arginine, lysine, histidine. Clear? Are you clear?
Clear, sir.
Good. Please mark the details.
Done, sir.
Have everyone completed writing them?
Yes, sir.
Good. So, amino acids present in the body of human can be divided into two types. What are the two types of amino acids?
Essential amino acids.
Essential amino acids and
Non-essential amino.
non-essential amino acids. Essential amino acids, read and see. They cannot be synthesized in the, what is it? Human body. Therefore, the only way to get it inside our body is eating food. Non-essential amino acids can be synthesized using whom it is synthesized?
Using essential.
Using essential amino acids. If you don't intake essential amino acid, you will be losing both essential amino acid and non-essential amino acid, because to make the non-essential amino acid, who are the ingredients? Who are the ingredients?
Essential
The
amino.
essential amino acids. If essential amino acids are not intaken, we will be lacking essential amino acid as well non-essential amino acid. And remember, essential amino acid, non-essential amino acid, both are equally important for metabolism. Both essential amino acids and non-essential amino acids are equally important for, what is it? equally important for
Metabolism.
metabolism. Metabolism wise, we don't divide them as essential and non-essential. Whether we can synthesize in our body or not. According to that only we have divided them as essential and what's it? Non-essential. Are you clear? Are
Yes,
you
sir.
clear? Please mark it down.
Done, sir.
Good. If you take the functions of protein, there are many functions done by protein. Your book is dividing those functions like this. What are the type of functions? Catalytic function. Second, making structures. Being a storage. Performing transportation. Being hormones or signaling molecule. Contractile function. Defensive function. What are the functions? What are the functions? Number one, catalytic function, catalyzing metabolic reactions. Second function,
Structural function.
structural protein mean making structures. Third one,
Storage function.
storage. That means protein can be stored in a place. Fourth one,
Transport.
transportation function. Fifth one, hormones. In brackets, write down signaling molecules. Hormones are home. Hormones are home.
Signaling molecule.
Signaling molecules. Next function,
Contractile or motor.
contractile, that is movement function. Finally, defensive function, that is giving us protection. Right? Who is the example for catalytic protein?
Any enzyme.
Any enzyme. In your O level, you studied many enzymes. Amylase, correct? Lipase, trypsin, chymotrypsin, pepsin. Studied no? Any enzyme, you can call it catalytic protein. Structural protein. Who are the examples given to you as structural protein?
Keratin.
Keratin and
Collagen.
collagen. Keratin and what is it? Collagen. I will. Now, keratin is not easy to show you. Okay, I will show you this. If you take the hand of this person, can you see that white color code-like structures? Can you see?
Yes, sir.
They are called by the name tendons. They are called by the name, what is it? Tendons. Because of these tendons, we can fold our fingers. Understood? If you hold one tendon by a forceps and pull it, your finger will be moving. Okay, they are rope-like structures fixed to the muscles. They are rope-like structures fixed to the, what is it? Muscles. Okay, these are models. They are not real. Can you see the tendons? Can you see the tendons? Yes. These tendons are made of the protein called collagen. What is the protein making these tendons? What is collagen? So, it is what we speak here. It is providing what it is providing.
Strength and support.
Then now say if you cut one person's tendon, now say for one finger, one tendon there, another finger, another tendon there. If you cut one tendon, then that finger will not work. Understood? Understood?
Yes, sir.
Good. Keratin is present in our skin. Keratin is present in our skin. In our skin, in the top layer, there is this protein called keratin. Because we have keratin, easily microorganisms cannot enter into our skin. Further, water loss from the skin will be very low. What are the importance of keratin in our skin? What are the importance of keratin in our skin?
Minimize water loss.
Minimizing water loss. Second one, preventing the entry of microorganisms. It is giving you protection. Third one, storage. Who are the examples given to you? One is this, one is this. What are the two example proteins for storage? Read it.
Valbumin.
Ovalbumin. O mean, my children, egg. Ova mean, what is it? Ovum, ova, ovum, singular, ova, plural. Ova mean egg. So how do you read that word? How do you read this word? The blue color one. Read it, read it. Read it,
Ovalbumin.
Ovalbumin. Ovalbumin is a storage protein in which component?
In egg.
In egg. In the egg, the white yolk is ovalbumin. Yellow yolk is lipid. White yolk only protein. So in the egg, ovalbumin, okay, it's a yellow white color or colorless protein, ovalbumin. What is the second protein? Storage protein. How do you read it? Read, will you?
Casein.
Casein. Casein. It is stored in which material?
In milk.
In milk. Okay. What are the transport proteins? Two transport proteins in syllabus.
Hemoglobin.
hemoglobin, serum,
Albumin.
albumin. Hemoglobin is transporting which components?
Oxygen and carbon dioxide.
Oxygen and carbon dioxide. Serum albumin is transporting which component?
Fatty acid.
Fatty acids. What are the hormones or signaling molecules that are proteins?
Insulin, glucose,
Insulin, second one.
glucose.
Read it. If you pronounce it, it will come. Don't worry about the pronunciation or anything. Just read it. What is the second one?
Glucagon.
Glucagon. Both of them are involving in one activity. What is that activity? Regulating blood glucose level. Insulin will decrease the blood glucose level. Glucagon will increase the blood glucose level. Both together, they will maintain the blood glucose level. What are the movement proteins or motor proteins?
Actinomyosin.
Actin and myosin. Actin is a motor protein, myosin is another motor protein. So they are helping in the contraction of muscle fibers. Because muscle fibers are contracting, we are able to move our body organs. And thereafter, protection proteins. What is the name of protection proteins? What is the name of protection proteins? Example.
Immunoglobulins.
Immunoglobulins. Another name for immunoglobulins? Antibodies. What is the work of antibodies? Eliminating foreign agents. Okay, in the upper level, you will study many different defensive proteins and many different foreign bodies. At the moment, foreign bodies mean what it comes to your mind is microbes. Are you clear?
Yes.
Right. With this one, protein portion is over with you. And today you are going to start to discuss regarding home. What is the heading, Asma?
Nucleic acid.
Nucleic acids. In your O level, in grade 10, very small amount of knowledge you gain regarding the nucleic acids. But in the advanced level, you have a bigger knowledge regarding nucleic acids. Clear? Right.
Yes, sir.
But we are just simply starting. What are the elements making nucleic acids? You are given. What are the elements making nucleic acids?
Carbon, nitrogen, phosphorus.
Carbon, hydrogen, oxygen, nitrogen. What is it? Phosphorus. Second point. Nucleic acids are polymers of whom? Polymers of whom?
Nucleotides.
Nucleotides. Now, if you search nucleic acid structure, okay, nucleic acid structure, right, I will choose this picture. Good. Here, see this one. This one piece is Putha, one nucleotide. This is another nucleotide. This is another nucleotide. This is another nucleotide. This is another one. 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 So do you agree? Many nucleotides are joined together to form a nucleic acid. Very simple idea. Not bigger ideas there. Ideas are simple. Do you agree? Many nucleotides are joined one after the other, one after the other, one after the other, and make a chain, and that chain is called by the name nucleic acid. Agree? Do you agree? Do you agree?
Yes, sir.
Good. And due to this reason, nucleic acids are also categorized under home. Biopolymers. Remember, biopolymers or macromolecules are three. One, it is polysaccharides. We already studied it. Second, proteins. We already studied it. Third, it is home.
Nucleic acid.
Nucleic acids. Who are the ones coming under biopolymers or macromolecules?
Polysaccharides.
Polysaccharides, proteins and nucleic acids. Clear?
Yes, sir.
Are you clear? Asma, just mark this one and keep. Polysaccharide, proteins.
Okay, sir.
After studying this one, you will further understand it. Biopolymers mean only three. In MCQ paper, if they ask you which one of the following are biopolymers mean or macromolecules mean, one will be polysaccharide, one will be protein, one will be home.
Okay, okay, sir.
Nucleic acids. Mark it down. Mark and keep. Okay. Have you marked it, Putha? Have you marked it?
Yes.
Right. What is the fourth point? Read the fourth point. Read the fourth point. Read, will you?
Even
Nucleotides are joined by
polyester bonds.
what bond?
Phosphodiester.
Phosphodiester bond. What is the bond? What is the bond? Right. Again, I am making a summary. Okay. If you take disaccharides and polysaccharides, okay, disaccharides and polysaccharides, what is the name of the bond present in them? What is the name of the bond?
Glycosidic bond.
Glycosidic bond. If you take lipids, what is the type of the bond present in them?
Ester bonds.
Ester bond, correct? If you take proteins, what kind of bonds present in them?
Polypeptide bond.
Peptide bond. Peptide bond. If you take nucleic acids, the bond present is called phosphodiester bond. Could you please write this summary and keep with you? Okay, what is the heading? Bonds in organic, biological organic molecule. Biological organic molecules. Okay.
Done, sir.
Good. We will further discuss regarding the phosphodiester bond. At the moment, just introducing you piece by piece. Okay. One more thing. One more thing. Okay. Let's go for this. Let's take this phosphodiester. What is it? Bond. Check whether in your tube this picture is given, Puta.
Yes.
Yes. Here, no need to take lots of details at the moment, just an idea. This is a nucleotide. Which one? This is a nucleotide. This is another nucleotide. Then how many nucleotides are shown to you here? How many nucleotides are shown to you here?
2.
2 nucleotides are shown to you here. Here, what is the number marked?
3.
3. That is indicating the third carbon. Which carbon?
Third carbon.
Third carbon. Here, what is the number marked?
5.
What is the meaning of it then?
Fifth carbon.
Fifth carbon. Phosphodiester bond is situated between the third carbon of one nucleotide sugar group and fifth carbon of the other nucleotide sugar group. That is the meaning. Later, we will fully discuss it. Just an idea for you to understand this point. Can you understand this point? Check, will you?
Yes, sir.
Yes. If you take a nucleotide, in a nucleotide, this is O level also you studied, but in the O level, very basic things you studied. There are 3 groups there. What are the groups there? Read it. Just look at the picture and tell me. What are the groups there? Asma, what are the groups?
Phosphate group.
Phosphate group.
Phosphate group,
Sugar.
sugar group,
Nitrogenous.
nitrogenous base group. My children, okay, this phosphate, this sugar group will be what kind of sugar group? What kind of sugar group, my children? It's a pentose sugar. Please mark it down. It's a pentose sugar. Okay, have you marked it?
Yes.
Yes, sir.
Right. So, in this picture, let's do a small thing. This place you mark number 1. This place you mark number 1. This is number 2. This is number 3. I am labeling the carbon number. This is number 4. And this is number, what is it? 5. Have you labeled it? Very simple, simple labeling.
Done, sir.
Right. My point is this. In this diagram, according to your numbering, the nitrogenous base, this whole thing is nitrogenous base, is attached to which carbon of the sugar?
First carbon.
First carbon of the sugar. Agree?
Yes, sir.
Right. This is attached to
Fifth carbon of the
fifth
sugar.
carbon of the sugar. Correct? Agree?
Yes, sir.
Mark those details. So you have to understand nitrogenous base is attached to the first carbon of the sugar and phosphate group is attached to the fifth carbon of the sugar.
Done, sir.
Right. One more point you mark and keep. Third carbon of the sugar involves in making which bond? Which bond? Which bond?
Phosphodiester bond.
Phosphodiester bond. Third carbon of the sugar involves in making phosphodiester bond.
Done, sir.
Good. So, if you take the nucleic acids, you know there are two types of nucleic acids. What are the two types of nucleic acids? One we call DNA, other one we call, what is it?
RNA.
RNA. Both are familiar ones. Yes or no? Both are familiar
Yes,
ones.
sir.
Yes. What is DNA standing for?
Deoxyribonucleic acid.
Deoxyribonucleic acid. What is RNA standing for?
Ribonucleic acid.
Ribonucleic acid. Why it is described by the word deoxyribo? Because in it, the pentose sugar called deoxyribose is there. Okay. What are the two? Okay. What are the pentose sugars? One pentose sugar is whom?
Deoxyribose.
Deoxyribose. Other pentose sugar is
Ribose.
ribose. Another one is there, ribulose. But that doesn't involve in making nucleic acids. So what are the pentose sugars involving in making nucleic acids? What are the pentose sugars?
Deoxyribose and ribose.
Deoxyribose and ribose. Okay, if deoxyribose is involving in making the nucleic acid, that is DNA.
DNA.
If ribose is involving, that is
RNA.
RNA. Are you clear?
Clear, sir.
Mark and keep this, pentose sugars. Others no need. Others already have studied. Others already have studied. Asma is new today, so in the midway now.
Okay, sir.
Right. Fine. Now coming to the nitrogen bases. These are the nitrogen bases. Okay. Right. Because they contain nitrogen and they show basic property, we call them nitrogen bases. Just let us read the names of these nitrogen bases. What are the nitrogen bases? Read this name.
Cytosine.
Cytosine. Read this name.
Thymine.
Thymine. Read this name.
Uracil.
Uracil. Read this name.
Guanine.
Guanine. Read this name.
Adenine.
Adenine. So altogether in a living cell, how many types of nitrogen bases are present?
5 types.
5 types are present. In the 5 types, cytosine, thymine, uracil, guanine, adenine. In the 5 types, okay, 3 of them are present in DNA and RNA. What are the 3 present in DNA and RNA?
Cytosine, guanine, adenine.
Cytosine, guanine, and
Adenine.
adenine. Only found in DNA. Which one?
Thymine.
Thymine. Only found in RNA.
Uracil.
Uracil. Correct? So, if you take DNA, what are the nitrogen bases found in the DNA altogether? Altogether, what are the nitrogen bases found in the DNA?
Cytosine, thymine,
Cytosine.
guanine, adenine.
Cytosine, thymine,
Thymine,
okay,
Guanine.
adenine, and what is it? Generally, we write like this. That is easy. AGCT. Adenine, guanine, cytosine, and what is it? Thymine. RNA. RNA. Adenine there, guanine there, cytosine there,
Uracil.
uracil.
uracil there. Correct? Understood?
Yes, sir.
Yes. If you take cytosine, thymine, uracil, in this one, how this is a ring. This is a ring structure. This is a ring structure. This is the ring structure. Check one by one. In cytosine, how many rings are there? One ring there. In thymine, one ring there.
One ring.
In uracil,
One ring.
one ring there. If one ring is there, we have to refer them by the word, what is it? Pyrimidine. What is the name?
Pyrimidine.
Pyrimidine. Who are the pyrimidines? Who are the pyrimidines?
Thymine,
Cytosine,
uracil.
thymine, uracil. If you take the next two, look at the diagram. How many rings are there?
Two rings.
Two rings. If two rings are there, how do you call it?
Purines.
Purines. Who is larger, purines or pyrimidines?
Purines.
Purines are larger. Look at cytosine. What are the elements making cytosine? Tell me. Carbon there.
Nitrogen.
Hydrogen.
Nitrogen.
Nitrogen.
Oxygen.
Oxygen. Agree?
Yes, sir.
Everyone agree with it?
Yes.
What about thymine? What are the elements making it?
Carbon, hydrogen,
Carbon.
oxygen, nitrogen.
Hydrogen, oxygen, nitrogen. Okay. What are the elements making uracil?
Carbon, hydrogen, oxygen, nitrogen.
Okay. What are the elements making guanine?
Carbon, hydrogen, nitrogen, oxygen.
So same thing repeating, correct?
Yes, sir.
What about adenine?
Carbon, hydrogen, oxygen, nitrogen.
Oxygen there?
Carbon, hydrogen, nitrogen.
Carbon,
Oxygen.
hydrogen, nitrogen. It is lacking
Oxygen.
oxygen. Clear? An oxygen
Yes, sir.
lacking nitrogen base. Who is it?
Adenine.
Adenine. Who are the purines?
Guanine and adenine.
Guanine and adenine. Who are the pyrimidines?
Cytosine, thymine, uracil.
Who are the large molecules?
Guanine.
Purines.
Purine. Okay. How many rings are making the purines?
Two rings.
Two rings. What are the nitrogen bases found in the DNA?
Adenine, guanine, cytosine, thymine.
RNA.
Adenine, guanine, cytosine, uracil.
So if these details are clear, we can move further. Please mark it down, these details on the diagram.
Done, sir.
Okay. Have everyone marked it?
Yes, sir.
Good, good, good. So, from that place, inshallah, we will continue. Okay. We'll meet inshallah. Assalamu alaikum.
Walaikum salam.
Thank you, sir.